Morning my dear students, welcome back to Sangu Home Classes. In the previous class we discussed about the nucleus. Today we are going to know about the remaining things like cytoplasm. What is mean by cytoplasm? Cytoplasm is a jelly-like substance which is present inside the cell. So the cytoplasm is containing number of cell organelles in it and it is bounded by plasma membrane. Okay. Observe the picture student. Here there is a cell, plant cell and also animal cell. See, when we observe in the animal cell, this is a cell membrane. Inside the cell membrane, all these organelles are located in the cytoplasm. Here there is a orange color fluid substance is there, no? So it means what? This is not always in orange color. So in this figure we are saying about the cytoplasm, that is in orange color, which is appearing in orange or a light yellowish color. So that is called as a cytoplasm. Yes, in this cytoplasm there is a endoplasmic reticulum, nucleus, mitochondria, Golgi apparatus and also some more cell organelles are present inside the cytoplasm. See, it is not a liquid, not a solid. This is a semi-liquid and semi-solid substance present in each and every cell. So, when we compare the prokaryotic and eukaryotic cells, so in the prokaryotic cells, these nuclear membrane which is bounded the nucleus is absent. And also cell organelles which are bounded by membranes, those are also absent. And except the ribosomes, all the cell organelles does not contain any membrane which is bounded the cell organelles, or it may be nucleus itself. Now look at your textbook, page number 5. So there is a small topic, that is protoplasm versus cytoplasm. What is mean by versus? So protoplasm is separate and cytoplasm is separate. Like that, in the earlier days the scientists are thinking like that. So after some days they are realized themselves what is happening. So see here, for a long time it was believed that the essence of life was stored in the fluid found inside the nucleus. So next generation, what are the characters are getting from older generations or parent to progeny. So those are all the characters are present inside they are all found inside the fluid itself. They are all thinking like that in the previous or in the earlier days. But after that, hence this was named protoplasm. So that is only they are called as a protoplasm, which means a life fluid. But after, when it became clear that the fluid is basically a medium in which various particles and also membranes float around, and that the functions of the cell actually takes place in these organelles. So it began to be understood that life resided in this organization. In particular, the materials inside and outside the nuclear membrane was differentiated after the discovery of nucleus. So what happens, students? So after the discovery of nucleus, the Robert Brown was the inventor the nucleus, no. So after the invention of nucleus, all the scientists could understand about the protoplasm and also nucleoplasm. What is protoplasm? The cytoplasm is nothing but protoplasm, and the nucleoplasm which is inside the nucleus is called a nucleoplasm. But what are the specific characters are coming from the generation which is from parents to progeny? So those characters are done by the responsibility of a DNA. So the DNA, where is it present? The DNA is present inside the nucleus. Okay, so the nucleus, the nuclear membrane, and inside the nuclear membrane there is a nucleoplasm, and nucleoplasm, it is containing the DNA. So the DNA only responsible for the genetic information which is coming from the parent to progeny, parent to young ones itself. So that is clear after the invention of nucleus. Do you understand? Yes. So hence protoplasm was renamed as a cytoplasm. So protoplasm, in the earlier days the scientists think protoplasm only responsible for all the characters, all the genetic information is present inside the protoplasm like that. But after the invention of nucleus, they concluded protoplasm is not responsible for the characters which are carrying from parent to progeny. So they concluded the protoplasm is nothing but it is a cytoplasm which is bound by the plasma membrane and also contain number of cell organelles which are responsible for the all the life processes which are involving in the living organism itself. So renamed as a cytoplasm. Then that is a cell fluid. Cytoplasm is also called as a cell fluid. It is only cell fluid. The fluid inside the nucleus came to be known as a nuclear fluid or nucleoplasm. So what did they confirmed at the last or after the invention of nucleus means plasma membrane is covering all the cytoplasm. In the middle of the cytoplasm there is a small dense ball-like structure is called nucleus, and nucleus is covered by nuclear membrane. Inside the nuclear membrane there is a nucleoplasm. Nucleoplasm only responsible for the genetic information which is moving or which is coming from old generation to new generation or from the parent to progeny itself. Do you understand, children? Yes. The next thing is cell organelles. Cell organelles are the very minute structures than the cell because they are present inside the cell. See, now let us discuss some important cell organelles like mitochondria, plastids, ribosomes, endoplasmic reticulum, Golgi apparatus, lysosomes and also vacuoles. They are important because they carry out very crucial functions in the cells as we know already. Yes, so in these there is some activities are done by the help of microscope. See what are we need to observe the mitochondria or any other cell organelles inside the cell. See, observing mitochondria. Let us do this activity with the onion peel. So if you take an onion peel, inside the onion peel there is a number of cells are located. So each cell contain number of cell organelles. So how to distinguish, how to observe the cell and how to observe the internal particles like cell organelles. See what do we need? See here, make a fresh solution of Jonas green bee in a beaker. So one type of solution we have to take it, that is a mix maximum about 200 milligrams. Next Jonas green bee in 100 ml of water. So mix చేసినటువంటి 200 mg of Jonas green bee and also 100 ml of water. So that is a solution. We should take the solution and also take a watch glass, pour some solution, put the onion peel in the solution and keep it for about half an hour. So what should we do now? We have to take a onion peel and keep in its solution itself for half an hour. Next, keep a piece of onion peel on the slide and wash thoroughly with water. After half an hour we have to take the onion peel, a piece of onion peel, and also keep it on a glass slide. Then wash thoroughly with water. We have to wash it thoroughly means we have to wash clearly. So cover the slide with a cover slip and observe it under microscope at a high magnification. It means what? We have to take a piece of onion peel, keep it on a slide. After that we have to cover it with a cover slip, and then we should going through the next observation with the help of a microscope. See here, If you observe and make a sketch of the same in your notebook, compare it with the given diagram. So if you observe under the microscope, the diagram will be appear, or the slide which is containing the piece of onion peel, that is appeared in this way, like this. So mitochondria in onion peel cell. So it will be appear like this. So here see what is there. We can do this activity by taking on other available material like leaves of cassia, thora, and also cheek cells. So cheek cells means it is an animal cell, and also cassia thora, it is a one more plant cell. So if you observe, we can observe the mitochondria along with the help of a cassia thora or cheek cells, it may be. Then you may have observed green oval or cylindrical grains scattered in the cytoplasm. These are called as a mitochondria. So how will they appear in the leaves? Means, so we can observe it is in a green color and which is in the shape of cylindrical grain like structure, and scattered means they are scattering inside the cytoplasm, means they are spreaded in the cytoplasm everywhere. See, if you observe the structure of a mitochondria, see it is a mitochondria, it is a cylindrical shape, it is in cylindrical shape, and mitochondria is a very small and also spherical or cylindrical organelle. We can call it as it is a small and a spherical or cylindrical organelle. Generally, every mitochondrion is 2 to 8 microns long. What is the length of mitochondria? 2 to 8 microns long and about 0.5 micron wide. What is the width? 0.5 microns width, and it is about 150 times smaller than the nucleus. So we know already a nucleus is a very minute structure. So more than this, it is very minute. How much? It is 150 times minute or small than the nucleus. So we are not able to see with clearly with the microscope also. There are about 100 to 150 mitochondria in each cell. See, these type of minute structures are present 100 to 150 in each and every cell. So when see under the compound microscope, the mitochondria appear as a oval or cylindrical dots in the cell. So if you observe under the compound microscope, how they are appeared in the shape of cylindrical shape, or it may be oval in shape. So the diagram of mitochondria shown in typical cell is a hypothetical diagram. So if you observe under this, to draw the mitochondria, it is very typical, very critical, very difficult. Okay. Electron microscope reveals their unique internal structure in great details. It means what? Through the compound microscope, we cannot draw the internal structure of mitochondria itself. If we use the electronic microscope, okay, if we use the electron microscope, we can see clearly and we can draw the each and every structure which is inside the mitochondria also. So information derived from the electron microscope tells us that the mitochondria are made up of double membrane wall. So how many membranes are present around the mitochondria? So it contains two types of membranes. One is a outer membrane and another is inner membrane. See here, this is the outer membrane and this is the inner membrane. See here, the inner membrane is protruding inside, foldings inside. We can observe some of the foldings are there. So these foldings which are inside, they are called as cristae. These inner foldings are called as a cristae. So in between the cristae, there is a fluid. There is a fluid content is there. So this fluid is called as a matrix. Fluid is called as matrix. What are the functions of mitochondria? Let us know. Mitochondria are responsible for cellular respiration. Means inside the cell there is a respiration process is going on. So that respiration is done by the mitochondria itself. A process through which the cell derives its energy to do work. So whatever the process is going on inside the mitochondria, it will be helpful to derive its energy to do the work of a person. సో ఏదైనా ఒక మనిషి కానీ ఒక జీవి కానీ ఎనర్జీకి రావాలి అంటే మైటోకాండ్రియా షుడ్ బి ప్రజెంట్. సో ఇఫ్ ద మైటోకాండ్రియా విల్ గివ్ ఎనర్జీ, దెన్ ఓన్లీ వీ కెన్ ఏబుల్ టు వర్క్. సో డూ యూ నో వన్ థింగ్? Even if you want to move our eyelids, it should be needs some energy. That energy will come from the each and every cell by the help of a mitochondria itself. So that's why the mitochondria is having another name, that is powerhouse of the cell. Why? The mitochondria is storing the energy. That's why they are called as, they are giving the energy for the cell and for the body. So that's why they are called as a powerhouse of the cell. They are called as powerhouse of the cell. What is the nickname of mitochondria? It is a powerhouse of the cell.